Ladies and gentlemen, welcome to session 4, Strengthening Global Preparedness through CEPI's 100 Days Missions. And welcome back to the World Bio Summit 2023. This session will focus on the crucial aspects of a rapid vaccine development and production in response to pandemics. So ladies and gentlemen, please stay with us until the very end of this session as well. In this session, we'll delve into the CEPI's 100-day mission and explore CEPI's collaboration with IVI. We'll also engage in diverse and meaningful presentations covering collaboration with Korea Disease Control and Prevention Agency and contribution for public health system with CEPI. And also, we'll be talking about engagement with academia and also the collaboration with industry. And each topic promises to offer valuable insights into the collaborative efforts and advancement in the field of vaccine development. So now, in order to start this session, it is my great honor and privilege that we can welcome a distinguished leader in the field of infectious disease innovation. Please join me in welcoming Dr. Yoon In-kyu, Director of CEPI. So please welcome him with a big round of applause.
Thank you very much, and welcome to session 4, the final session of the World Bio Summit. Thanks, of course, to the co-hosts of the World Bio Summit, the Ministry of Health and Welfare, and the WHO for a very successful meeting. And thank you to the International Vaccine Institute for co-sponsoring session 4 with CEPI. And this session is entitled Strengthening Global Preparedness Through the 100 Days Mission. CEPI's 100 Days Mission states vaccines should be ready for initial authorization and manufacturing at scale within 100 days of recognition of a pandemic pathogen. A key tenet of the 100 Days Mission is that neither CEPI nor any single organization, nor any single country can prepare for the next pandemic alone. So today, in this session, we'll highlight some of the key collaborations that will make the 100 Days Mission possible. Collaborations with governments, with international organizations, NGOs, industry, and academia from throughout the world. In fact, a large part of why we are all here at this summit is to seek out and expand these necessary partnerships. So I hope we can move forward with this ambition, and I hope you're looking forward to the session as much as I am. Thank you very much.
Thank you very much. Let's give a big round of applause. Thank you for that wonderful opening remarks. And now we'll delve into a panel discussion, and we would like to welcome our esteemed panelists. Once I mention their name, please welcome them with a big round of applause. First and foremost, we'd like to welcome Honorable Ms. Jane Halton. Please come up to the stage. Let's welcome her with a big round of applause. Thank you very much for your presence today. And next, I'm honored to invite Honorable Dr. Jerome Kim. Please come up to the stage. Let's welcome him with a big round of applause. And we also have Honorable Dr. Toronto Monroe. Please come up to the stage. Thank you. Next, please join me in welcoming Honorable Emmanuel Montemoli. Please come up to the stage. Let's welcome him. Thank you. Next, we have esteemed Shadik Akasha. Please come up to the stage. Let's welcome him. Thank you for your presentation. Thank you. And next, we would like to welcome Honorable Park Jin-sun up to the stage. Let's welcome her with a big round of applause. And we're also joined by esteemed panel Lee Gi-eun. Please come up to the stage. Let's welcome her with a big round of applause. Thank you once again. And ladies and gentlemen, last but not least, this session will be moderated by Ms. Kate Kelland, a chief science writer at the Coalition for Epidemic Disease Innovation. So please welcome her with a big round of applause. So she is chief science writer at the Coalition for Epidemic Preparedness Innovation. And for a brief introduction of our moderator, Ms. Kate Kelland is the author of the 2021 book titled Disease X, The 100 Days Mission to End Pandemics, and former global health correspondent for the Reuters news agency as well. Once again, let's welcome her with a big round of applause.
Thank you very much, and good afternoon to all of you, and thank you very much to all of our speakers for joining us today for what promises to be an enlightening and invigorating discussion on what it will take to be able to execute a successful 100 days mission when a new Disease X pandemic threat emerges. My name is Kate Kelland. I'm the Chief Scientific Writer at CEPI, the Coalition for Epidemic Preparedness Innovations, and also the author of a book called Disease X, which is about the 100 Days mission and about how we can end pandemics. I'm really looking forward to moderating the session today. And I'd like to start, please, by inviting Professor Jane Halton, who is the chair of CEPI's board, to talk to us about the mission, about what it means, and about what it will take to get mission ready. Jane, can I ask you to come to the podium? Thank you.
Good afternoon, ladies and gentlemen. It's very good to see so many people still bright-eyed and bushy-tailed at the end of a very busy couple of days. And it's my particular pleasure to be here in Seoul on what is actually outside a beautiful autumn's day. And of course, the sunshine we see outside is a very welcome flicker of light at a time when there is so much darkness enveloping the world. And it's also an honour to be asked to speak to you this afternoon and to be able to have met so many of you in person and to discuss how we can work together in this era of crisis, threat, and opportunity. It's now been more than 6 months since the World Health Organization declared that the COVID-19 pandemic, or at least, of course, its acute and deadly phase, is over. And around the world, while the disease is still with us, COVID case numbers, compared to the midst of the crisis, are waning and death rates are decreasing. Although, of course, I do note that in many parts of the world, Australia included, we are seeing yet another wave. Of course, this waning of cases compared to the peak of the pandemic is something that we should welcome and celebrate. Unfortunately, however, these dwindling figures also mean that our memories will begin to fade and pain of the loss that has been experienced by so many will begin to ease. And alongside the fading of those painful memories, our desire to forget and to move on, I think, is real. And of course, that's understandable. Governments and populations around the world desperately want to get back to life as it was, even if it's a sort of new normal. They have other multiple complex demands on their time, on their resources, their effort, their money, and indeed their empathy. And as I think many of us would know as we turn on our televisions, war and conflict dominate the headlines. Extreme weather events dominate our forecasts, and of course, economic and financial downturns dominate our everyday lives and our worries. So it's no exaggeration to say we're living in an era of polycrisis. Yet today, as governments seek to re-establish a version of the new normal, after the COVID-19 pandemic. Commitments have been stripped back in some areas of global health and humanitarian crisis response. This, to me, is deeply worrying. It's evocative, sadly, of so many previous periods of human history. We've seen this with events like the 1918 flu pandemic, or the 2009 H1N1 swine flu pandemic, the deadly emergence of MERS, particularly, of course, in this country in 2012, and, of course, the devastating Ebola epidemic of 2014, becoming forgotten pandemics, and surprisingly, very quickly forgotten. When novel viruses spill over and cause disease, death and destruction in our world, we are at first horrified, but then we're very swift to begin to forget and to become distracted. We fall into, as I said this morning, the cycle of panic and neglect. This behaviour is dangerous. It should be avoided. And instead, we need to recognise that every time we experience an event like SARS or swine flu or MERS, we have to learn from these events. Korea has demonstrated with great clarity in its praiseworthy response to the emergence of SARS-CoV-2. It had learned. And I've said this to government officials and politicians here today. We can see the impact that something like MERS had on the response when yet another coronavirus spilled over. And of course, as with so many other viruses, it hitched a ride on a Korean business traveller and sparked an outbreak many thousands of miles away, right here in Seoul. The MERS outbreak that erupted in Korea was the largest outside the Arabian Peninsula. And while it only affected, and I use the word only with some caution, around 300 people, which some people would say is very few compared to flu or COVID-19, it was devastating to the economy, to the Korean tourism industry, and to the individuals and their families who were affected. So when the next novel coronavirus emerged in the shape of COVID-19, Korea had learned a lot. This country's response to the pandemic has been studied and documented with some admiration, and it is often held up as an exemplar to others. That's because Korea did not forget and neglect. It learned, and what it learned best of all was how to prepare better. So it's imperative that we keep the COVID-19 pandemic and the death and destruction it caused in our minds as global health security leaders, and of course in the public consciousness, so that we minimise the risk that the mistakes of the past will be repeated yet again. That's why this World Bioconference and other gatherings like it in different regions of the world are so vitally important and, of course, so timely. Now is not the time to put pandemics behind us and to move on. Now is not the time to let our collective attention be completely consumed by conflict and violence and climate crisis. We know that pandemics and epidemic threats are unpredictable. The one thing we can predict is that another outbreak or epidemic will emerge and threaten to explode into a pandemic. So now is the time to prepare for what we know will, without doubt, emerge again. A future infectious disease threat is an inevitability, and the time to prepare for the world is now, and how we can respond is something we need to think about now. I might underscore that if I had heard one more time in some media interview someone describing the COVID-19 pandemic as a once-in-a-100-year occurrence, I probably would have screamed or throttled somebody. We don't know when this next pandemic will come. We simply know that it will. Many of you, I expect, will have heard talk of what we call the 100-Day Mission, and I commend Kate's book to you. It is excellent. But I hope, even if you haven't heard of the 100-Day Mission, that you will have heard of my organisation, CEPI. the Coalition for Epidemic Preparedness Innovations, which is a bit of a tortured name, which is why we just call it CEPI. And we are the key architect of the first 100-day mission. And it's an idea that has been embraced by leaders of the G7, the G20, and as a plan for the world. And of course, by Korea, who have both a 100-day and a 200-day mission. And it's about dealing with any conceivable viral threat, new or re-emerging, within as little as 3 months from the recognition of that threat. The 100-day mission is ambitious. Few would argue with that, but it's also, in my view, realistic. It's unarguable as a vision that can help us towards a safer future. The faster an effective vaccine is developed and deployed, the faster people can be protected from infection or death from a viral pathogen, and the faster an incipient pandemic can be contained and controlled, the better off we will be. In the case of COVID-19, a 100-day timeline would have seen a vaccine ready, and think about this. It would have seen a vaccine ready for use in mid-April 2020 instead of December 2020. It could have saved millions and millions of lives and trillions of dollars. So what will it take to be ready to embark on a successful 100-day mission? How do we get mission ready? With the help of its partners, CEPI has been thinking in great detail about this. In November of last year, we published a report entitled, "What Will It Take?" And that's about how governments and the wider community can best prepare to prevent the next potential pandemic. Among the most vital elements of better preparedness identified in that report is greater cooperation between researchers, vaccine developers and regulators, between governments, public health authorities and funders. Many of the partnerships CEPI has forged here in Korea are fine examples of that. We've teamed up with SK Bioscience and with IVI to work on the development of a whole new vaccine platform technologies and priority pathogens that can teach us how to do these things better. I'm very pleased, as I've already said, about the 100-day mission here in Korea, and the Minister and his colleagues have set out a response plan to deal with these issues. So we commend that, and we would strongly argue others should do the same. I'm also particularly pleased about the key areas in the Korean National Pandemic Preparedness and Response Plan, which align very closely with CEPI 2.0, the plan we have for the future. And that emphasises innovation and investment in preparedness research and development, surveillance and prevention, and on infrastructure expansion, and of course in this country commits resources and expertise to the mission. With its world-leading life sciences industry and its commitment to pandemic preparedness, both at home and abroad, I've no doubt Korea will play an absolutely pivotal role in this agenda and global health security. The Korean government has also doubled down on its commitment to this global mission with a generous pledge for 2023 of $24 million, and we're very grateful for that support. And that's come at a critical time for the work that we are developing going forward, and we actually commend them for their dedication to tackling the kind of threat that we all know emerging diseases, infectious diseases, continue to be. We know that developing safe and effective vaccines in 100 days can only be truly effective in thwarting a future pandemic if the production of those vaccines get to the people who need protection as quickly as possible. That means they can be produced at scale and deployed wherever they are needed. And of course, that means at the epicenter of any outbreak where there is the highest risk and those at highest risk of its imminent spread. The inequity of vaccine distribution that not only increased the death toll of COVID-19, but also tightened and prolonged its deadly hold on the world, was essentially due to a concentration of vaccine manufacturing capabilities in just a handful of areas and countries around the world. This must change. So to complement the 100-day mission, the thing we also must achieve, if we are truly to protect the world from future epidemics and pandemics, is the expansion and more equitable global distribution of vaccine manufacturing. Every region of the world should be able to manufacture vaccines for their own use, and every region should have access to rapid response platforms that will be our first line of defence. And again, I'm pleased to see that Korea is taking a leading role in this effort, and last year established a global manufacturing workforce training hub with the support of the WHO. And with this very clear example, Korea has indicated that it is embedding health security as a core component of its foreign policy and its development agenda, and I commend them for that. It's rapid and it's tangible progress, and we wish to see this kind of progress in other countries and reflected around the world. I began today talking about the rare and welcome light this visit to Korea and this summit is bringing to these rather dark times. I'd like to end by emphasising that amid the violence and terror, the disease outbreaks and extreme weather events, because I don't want you to go home feeling gloomy, there is good reason for optimism. The political and scientific endeavours we see are, I believe, focusing on pandemic prevention. Now, do they go far enough for many of us? Possibly not. Are they important stepping stones? Yes, they are. We've seen a declaration at the UN General Assembly that recognises the need for concerted effort. We know that there are discussions going on globally in respect of hopefully a treaty. We are learning about the collaboration we need to actively implement if we are to be prepared. And of course, more recently, and I'm sure you had a quiet cheer when you saw this, as did I, the vaccine technology pioneers Katalin Karikó and Drew Weissman won the Nobel Prize in Medicine for their work on mRNA, that groundbreaking science that was so fundamental to our response. Their work and this recognition, I think, is testament to the importance of sustainable investment in basic science to bring about real-world transformation. And we are learning about the science we need to prepare. The next disease X may or may not be deadly, highly contagious, and a threat to our way of life. But one thing is certain: to prevent a world which spirals out of control in the next pandemic, we have to be ready. And we now know that just because we can't predict precisely when it'll be or exactly what it will be, that doesn't mean we can't plan for it. With the learnings that we had from COVID-19 and the leadership of national and international experts like the speakers we're going to hear from in this session, and from all of you in this room, we have a once-in-a-generation opportunity to change the way the world prepares for pandemics and gets mission ready. I am optimistic. We can learn, we can prepare better, we can be ready. With government prioritisation of pandemic preparedness and equity, with sustained financing in vaccine research and development, manufacturing and outbreak response, and most importantly, with international cooperation and goodwill, we can build a better future free of pandemics. Thank you.
Can I use this one? Yes, thank you. Thank you very much, Professor Houghton, for that very insightful and inspiring assessment. I'd like to turn now to Dr. Jerome Kim, who is the Director General of the International Vaccine Institute, or IVI, and a long-time supporter and partner of CEPI in the global effort to reduce the risk posed by infectious disease epidemics. Welcome, Jerome, and the floor is yours.
Thank you. My name is Jerome Kim. I'm the Director General of the International Vaccine Institute while our slides are getting ready. IVI is a small international organization, actually the first international organization to be headquartered in Korea. With 39 member states and WHO as state parties, we have a mission to discover, develop, and deliver safe, effective, and affordable vaccines for global health. IVI works in over 30 countries in over 50 sites around the world and has, let's see, sorry, we're still waiting for slides. No? Okay, works in several sites, over 50 sites around the world, bringing innovative vaccine technology to companies, countries, and people. The IVI oral cholera vaccine was developed with a grant from the Bill and Melinda Gates Foundation, pre-qualified in 2011, technology transferred to several companies around the world, and is roughly 90% of the current global stockpile of oral cholera vaccine. We are engaged right now with a very important technology transfer with BioVac in South Africa, and I'm happy to say that the first stage of that technology transfer has occurred successfully, and we're waiting for BioVac to begin the process of scaling up. It's a very exciting opportunity, both for IVI and for BioVac, to bring new vaccine technology manufactured from scratch in South Africa to global health. Okay. All right, so I guess the first thing to talk about, since we don't have the slides, is the work that IVI and CEPI are doing together to advance pandemic preparedness. The first has to do with some scientific work that IVI did with some funding from CEPI around the preparation of a standardized serum for Middle East respiratory syndrome.
Sorry, Jerome, would you like us to help you out and let another speaker go first, and then would that be better?
I think that would be, sorry.
So while we get your slides sorted out, can I please welcome Dr. Gyun Ri, who is Director General of Korea's Disease Control and Prevention Agency. Dr. Ri, I hope your slides are ready. We look forward to hearing your perspective. Thank you.
Okay, thank you. Good afternoon. I'm Gyun Ri, the Director General of the Center for Vaccine Research at Korea Disease Control and Prevention Agency, KDCA. It's a pleasure to be given this opportunity to speak to you all. This is my presentation index. First, I will briefly introduce you about the Center for Vaccine Research at the KDCA. The Center for Vaccine Research is comprised of 4 divisions with 79 scientists, and the center is part of the National Institute of Infectious Diseases, which is under the Korea NIH. And as you can see here, KDCA is on top of all institutes and centers. Then, what does our center do? The mission of the center is the localization of public vaccines and securing a rapid support system for vaccine development. For the mission, we are establishing a vaccine development policy and roadmap. Moreover, the center is covering all the process of vaccine development from discovering candidate to a development stage, including preclinical and clinical trials. On one hand, the center is developing its own public vaccines, and on the other hand, it is providing support for private sectors. In order to ensure successful vaccine development, the center is in domestic cooperation with industrial, academic, research, and research institute and public sectors. On the international front, the center is working with USNIAID, Moderna, IVI, and CEPI as well. These are the vaccines that our center is developing on its own. They are mostly what the private sector avoid to develop due to no market demands, but are expected to tackle unsolved diseases and keep public health safety. Among them are next-generation tuberculosis vaccine, a human adenovirus type 55 vaccine for pneumonia outbreaks in Korean military bases, and third-generation smallpox and anthrax vaccines that are designed to cope with bioterrorism. Regarding anthrax, we submitted to an NDA for an anthrax vaccine to Korea MFDS, and we are waiting for its decision. Then we will move on to Korea's COVID-19 vaccine development. Korea's first licensed COVID-19 vaccine is SKYCoV-1, developed by SK Bioscience. It's a protein vaccine. The vaccine research began in March 2020 and was granted approval in June 2022. It took approximately 25 months. In addition, U-Biologics protein-based COVID-19 vaccine was granted export approval by MFDS as well. In the meantime, 2 mRNA vaccines delivered encouraging outcomes by entering clinical trials. These vaccines have been supported by Korea mRNA Vaccine Initiative. This achievement was in part due to the pan-government support. In June 2020, a pan-government support committee for COVID-19 therapeutics and vaccines was established as per a prime ministerial decree. The committee provided government policy support with the aim of developing therapeutics by 2020 and vaccines by 2021. This table shows the pan-government collaboration to accelerate COVID-19 vaccine development. We, KDCA, participated in this collaboration through conducting preclinical trials and clinical sample analysis with IVI. We were also responsible for providing government budget support to developers, along with the Ministry of Health and Welfare. Through all these activities, we KDCA have learned 2 lessons. Lesson number 1, we should prepare a rapid vaccine development system with core technology before our next pandemic occurs. Lesson number 2, pan-government support, continuous budget funding, and international cooperation with other entities are all essential. Now I will elaborate on Korea's vaccine development strategy against pandemics. In May 2023, KDCA announced the National Pandemic Preparedness and Response Plan based on our experience and lessons learned during the COVID-19 pandemic. At the same time, as indicated in the plan's core task number 10, KDCA is reforming its R&D system to expedite the development of vaccines and therapeutics together with its development strategy. This is our vaccine development strategy against future pandemics. A key takeaway here is a segmented approach. Before an outbreak, we are gearing ourselves up by establishing a rapid vaccine development system. When the outbreak occurs, we respond to it by developing vaccines within 100 to 200 days, similar to CEPI's plan. And then we are making continuous effort for evaluation and complementation. Step 1 is to establish a rapid vaccine development system before a pandemic. To this end, we have to keep 3 things in mind. Number 1, we select priority pathogens. Number 2, we secure new core technologies for rapid vaccine development. Number 3, we create a vaccine library. Number 1 is selecting priority pathogens. Korea selected its 9 pathogens by referring to those of WHO, CEPI, and the USNIAID. We also considered our domestic situation and advice from internal and external experts. Then I will sketch out our vaccine development plan against the 9 priority pathogens. First, we will take a two-track approach to the pathogens. As for high-risk pathogens with high prevalence rates in Korea, we will develop our own vaccines with a view to be given NDA's approval. And for the pathogens with low prevalence rates, we will work in global coordination with the aim of completing phase 1 clinical trials and reserving the vaccine candidate in our vaccine library. Number 2 is securing core technologies such as mRNA. mRNA technology ensures the most rapid response when a pandemic hits. To secure such technology, we will adopt technology from abroad in the short term. In the long term, we are working towards creating a Korean version of mRNA vaccine platform with continuous technology support. Number 3 is creating a vaccine library. You can think of this as a library collecting and reserving prototype vaccines that are all validated through phase 1 or 2 clinical trials. With these prototype vaccines, we will be able to conduct the rest of clinical trials rapidly and produce the vaccines within 100 to 200 days. With this in mind, KDCA has begun to found KAVAD, the Korea Advanced Center for Vaccine Development. For KAVAD, one research facility building in a Gyeongbuk Bioindustrial Complex will be established, and it will be in full operation in 2028. KAVAD will be entrusted with two big projects. Number 1, AI-based antigen designing, and number 2, establishing and operating a vaccine library. 9 priority pathogens will be our first main target for this. We expect that Korea's ability to respond to pandemics will be enhanced through the foundation of KAVAD. We will move on to step 2, response. The essence of response step is to complete the vaccine development within 100 or 200 days. In order to realize 100 to 200 days mission, we need 2 more factors. First, we need multi-institutional and pan-government cooperation to prepare a full-fledged support system. Second, preclinical, clinical trial, and laboratory networks should be strengthened in order to shorten the clinical trial period. When a pandemic occurs and one of our vaccine prototypes prepared through phase 1 or 2 clinical trials happens to target the same pandemic pathogens, we will immediately move on to a phase 3 clinical trial and produce the vaccine in 100 days, skipping earlier stage testing. However, if the prototype vaccines happen to target a similar pathogen, not the same pathogen, we will develop and produce the vaccine in 200 days by adopting already secured pathogen and vaccine information. MFDS's regulatory review is not included in the time period. I will briefly talk about step 3, evaluation and complementation. Step 3 is a stage to continuously validate and improve already approved vaccines. Efficacy evaluation in clinical trials, evaluation of neutralizing activity against variants, and long-term immunogenicity observation are included in this step. The evaluation and observation results will be reflected in policy for vaccine improvement, immunization, and epidemic prevention and control going forward. For all this, we are planning to facilitate support for private sectors with our BL2, 3, and 4 facilities, as well as the National Culture Collection for Pathogens at KDCA. Next is our future plan. As mentioned earlier, the National Pandemic Preparedness and Response Plan was announced last May. Now we are drawing up a detailed action plan and trying to secure more budget. With our plan, we will lay a legal groundwork, accelerating mRNA vaccine technology, and generating a vaccine library. Moreover, we will be in continuous cooperation and collaboration with other entities. This is my last slide. Korea wants to join forces to contribute to global health safety. With this resolve, at the G7 summit held in last May, the Korean government pledged to contribute to $24 million to CEPI, following our initial promise to provide $3 million. We hope that this will be a catalyst to move forward. Then I would like to mention potential collaboration areas with CEPI. We, KDCA, have started to talk with CEPI since last September. The first collaboration area can be vaccine development against viruses of mutual interest by forming a Korean consortium. KDCA and the private sector and possibly IVI are all expected to join the consortium going forward. A Lassa virus vaccine may be the very first product of these collaborations. The next cooperation area would be our participation in CEPI's vaccine library development project through our CAVAD. The talks with CEPI are currently in a nascent stage, and we are expected to begin to see tangible outcomes sometime soon. Distinguished guests, allow me to conclude by saying that Korea will be able to effectively prepare for and respond to future pandemics with our plan in place. Thank you.
Thank you very much indeed. Fascinating to see things from Korea's perspective and to look forward to future collaborations as well. So, Dr. Kim, I think your slides are ready now, so please re-welcome Dr. Jerome Kim to the podium.
I'm sorry again, but distinguished panel members, esteemed colleagues, good afternoon. I'm really going to talk about the collaboration that IVI is currently doing with the Coalition for Epidemic Preparedness Innovations, or CEPI, and then move on very briefly at the end to the 100-day mission and the things that we hope to accomplish looking forward. So I've gone over this slide already about IVI's mission to discover, develop, and deliver safe, effective, and affordable vaccines for global health. The way we do this is really almost a triangle with vaccine development, that is from the laboratory all the way through approval, uptake, formation of stockpiles, and considerations of policy. Understanding the burden and the impact, so understanding what diseases exist and what their burden is, and then looking at the vaccines that are developed, because in the end, vaccines should be cost-effective and impactful. And finally, a big component of what IVI does is around tech transfer and human capacity development through the Global Training Hub in biomanufacturing, but also through 12 and potentially now a 13th tech transfer of 6 different vaccines to developing country manufacturers around the world. This is where IVI works. You can see Latin America, Sub-Saharan Africa, South Asia, and Southeast Asia. These are clinical sites. We have a headquarters here. We have a regional office in Stockholm and a country office in Austria, with a consideration now for opening up an African office just approved by our Board of Trustees. So these are the areas where we have engaged currently with CEPI. First, in laboratory research, the establishment of a MERS reference standard serum with NIBSC. This has actually been, I think, very useful because I think many of us recognized actually pretty early on in the COVID pandemic that the absence of standardized serum really was going to impair our ability to compare between vaccines and being able to do this for other diseases, particularly diseases on the priority list for CEPI and WHO and others, for the government of Korea as well, is going to be critical in being able to standardize platforms between vaccines or between companies. And just to say, companies don't always necessarily like to be compared one against the other. So having standardized serum is very helpful in that regard. The second part of this is around epidemiology. And you know, I think that engaging manufacturers from all over the world has been one of the biggest tasks that CEPI faces. Because in the end, these vaccine solutions are helpful if we can, as we did during COVID-19, manufacture nearly 12 billion doses in 1 year. But to do that, you need to engage industry. And when you think about where the vaccines for global health come from, India and China are two major vaccine manufacturers. So we think it was very important for CEPI to fund this work, looking at the effectiveness of either Sinopharm or Janssen vaccine boosters to a Sinopharm primary dose to see what the difference in effectiveness could be between the two vaccines. It was also a good opportunity for IVI and CEPI to engage with Sinopharm, which is the largest manufacturer of vaccines in China. The second part is a recently announced opportunity with CEPI around preparation of phase 3 trial sites for Lassa fever. Again, as we think about pandemic preparedness, readiness of sites, getting countries ready to be able to review a protocol, to be able to consider the ethical implications of a rapid testing of a new vaccine, all of these things are going to be important, and familiarizing countries, IRBs, regulatory agencies with these new vaccines is important if we want to be able to have an emergency use listing of vaccine in 100 days. And you'll see that on a subsequent slide. So epidemiology was very important, and the preparation of clinical sites, again, fits in very well with CEPI's new mission to make vaccines available within 100 days. The third aspect of the work that we've done focuses around COVID-19 vaccine development. And IVI has been working with CEPI first for, actually with the KNIH and the Vaccine Research Center doing mix and match clinical studies of different COVID-19 vaccines in different locations around the world, including Pakistan and Madagascar, and then working with SK Bioscience in the SKY-CoV-1 vaccine testing outside of Korea, and then doing the testing of the neutralizing antibody and pseudovirus assays in Korea in conjunction with the KNIH. A very important ability of IVI to work with a partner government, as we have in Korea and hopefully can do elsewhere around the world. In terms of non-COVID vaccine development, we are working on a chikungunya vaccine with Bharat, funded by CEPI. That vaccine has entered a phase where it should enter phase 3 trial testing, hopefully early next year. That vaccine looks very interesting. It appears to protect monkeys against infection. Serum from people who've been vaccinated protects monkeys against chikungunya challenge, which is a useful surrogate for effectiveness of a vaccine where the actual efficacy testing may be difficult. And as we think with CEPI around these diseases, we're going to have to realize that it may not always be possible to do a phase 3 trial. So coming up with the mechanism for engagement of countries and other organizations around approval and use of vaccines for which phase 3 efficacy data is not available becomes a really critical aspect of the 100-day mission. The final part here is the is working with manufacturers in Korea and elsewhere to encourage them to work with CEPI. And you know, the engagement with Sinopharm started in early 2020, although it's taken a while to get full engagement and funding available through CEPI. Engagement of manufacturers, whether it's Bharat or Sinopharm or SK Bioscience, which is already an implementing partner of CEPI, which made it easy, is very important because in the end, it's these manufacturers who are going to make the product that will be used to vaccinate people around the world. We also work on WHO priority diseases that are not necessarily funded by CEPI. I've shown some of them here. MERS work with Inovio using a DNA vaccine. A Lassa fever vaccine, this is our first collaboration successful with the Hilleman Labs In Singapore, it's the measles-based Lassa fever vaccine funded by Innovate UK. And then finally, a CCHF vaccine in collaboration, that's Congo Crimean hemorrhagic fever vaccine in collaboration with the Karolinska and funded by the European Union. So IVI is continuing to work, and whether this work actually then merges into work that CEPI supports in the end, I think is one of those, is very important, because in the end, we have to find manufacturers. Manufacturers are going to want some aspect of guarantee of assurance that funding can be provided to take a vaccine to a certain level. And I think one of the things that we found is, and I think people around the room will agree, when you have the funding to go to the point where you need to, so that you can think about what the use of the vaccine is in the end, it's very helpful, because it allows you to negotiate not only with companies, but with regulatory agencies and other parts of governments in order to convince them that testing the vaccine is something that is worth doing. There are other things that IVI does that are not necessarily on the CEPI roadmap. Some of them are, but really get to some of the questions that we would like to address. The first is around the virus library, and I think the Korean government is very interested in that, so we will continue the work to work with KNIH on the virus library and the Vaccine Research Center on the virus library. Interesting new work on Nipah virus vaccines, again, in several different iterations in different ways, but this is a regionally important vaccine in Asia. One of the other aspects of this is that the microarray patches, which are now on the prioritization list for Gavi, might be a very interesting way to deliver vaccines that need to be heat stable and stored for extended periods at high temperatures. The other thing is that if you had a vaccine delivery system that targeted dendritic cells or other skin-related antigen-presenting cells, you might be able, using a patch, to be able to generate immune responses that are designed to target cells that are going to be important in generating immune responses. Dr. Ri has already talked about the work that we're doing with the Korean government on SFTS and hantavirus vaccines. Rift Valley fever is something that we've been very interested in and have had discussions already with companies like Afrigen that we hope to advance to further preclinical and clinical work. The Global Training Hub in Biomanufacturing, I think many people have heard about that. That's the panel, the last two panels, the red and the blue panels, talking about the roughly 600 people who've been through the GTHB, which is a strong collaboration between the Ministry of Health and Welfare of Korea and the World Health Organization. IVI is proud to be one of their partners in educating people, and I think it's actually now 2 months early, but we weren't going to do hands-on training until next year, but with funding from KWTRP, Khmer Wellcome Trust Research Program, which is based in Kilifi, and also with the Inter-American Development Bank, we are able to start the hands-on training in biomanufacturing this month, which I think is acceleration, but a very useful acceleration. The final aspect here, and actually it was mentioned before, IVI really strongly believes in equity. We wouldn't be doing tech transfers of technology to developing country vaccine manufacturers. We wouldn't work on diseases that are unincentivized if we didn't believe fundamentally in equity and access. AVA, the African Vaccine Alliance, is IVI's attempt to work with organizations in Africa to build an R&D ecosystem that is robust, that will support sustainable vaccine manufacturing. This doesn't mean building a factory, a vaccine manufacturing site. It means being able to, it means working with organizations in Africa to train people to do the things that IVI does, to secure funding, to work in the laboratory, to develop the clinical assays and validate them, to do the regulated human clinical trials to get vaccines approved, to work with regulatory agencies to get those vaccines approved, to work with WHO and Gavi to ensure that recommendations for the vaccine are made, that the vaccine is pre-qualified, and the vaccine can be purchased and used. Thinking end-to-end is a critical part of vaccine development, and we think that AVA is that. The one thing that distinguishes this from other activities is we are proposing to set up an organization that is separate from IVI. that will exist in Africa and be used and for the purpose of developing vaccines in Africa for Africa, and hopefully then manufactured in Africa using some of the manufacturing sites that the world has committed $5 billion to put up. But creating the ecosystem that will support sustainable manufacturing is the point of the AVA. So we are an implementing partner to CEPI, and we're very happy to do it. It covers a very broad range of topics along the entire value chain, along the areas where CEPI and IVI have a lot of alignment and common activities, from clinical development to manufacturing and technology transfer, the use of innovative technologies like patches, to work on standards like the MERS standardized serum, to work on vaccine pharmacovigilance, because for many of these vaccines, when we're doing rapid testing, we don't know the long-term safety issues. To work on stakeholder engagement, including other funders. So you can see that for some of the non-CEPI funded activities, we're working with some very unusual funders, and you'll see if you look at some of the other things in the IVI portfolio, we work with groups like Open Philanthropy, the Wellcome Trust, the Gates Foundation, and others, in order to secure funding to move global health vaccines forward. And we're proud to be able to work with CEPI to secure some of those additional partnerships, including what we announced last week, which is a partnership with the Hong Kong Jockey Club Global Health Institute, which is a collaboration between IVI, Hong Kong University, and Cambridge University. This is a $65 million effort to bring additional vaccine process development activity to the IVI capabilities folder, which will allow us to bring forward more valuable products that can be tech transferred in the future. So the 100-day mission. And the 100-day mission has been talked about before. I think, as was mentioned by Dr. Rhee, it's really about preparation. You have to have funding, which means that someone has to be thinking that funding of a certain envelope needs to be available to think end-to-end about the vaccine development, purchasing, and use. We have to prepare regulators for expedited approval. We have to be able to move forward with assessments, and it takes a while to validate assays. So as many people have mentioned, this is preparation. This is preparing those assays for the time when they'll be needed when we're testing the vaccines that are in the cabinet, waiting for potential use. We will need the platforms that include prototype vaccines, prototype processes and production, ready and able to move quickly. And I think you heard what Korea is doing. Other countries around the world are doing the same thing. Participatory practice. We don't often talk about this, but try taking a brand new vaccine into a country that thinks that you might be using them as guinea pigs. We need to be able to do the right things in order to engage populations, talk with people, and get them interested and engaged in helping us understand the importance of these vaccines. And finally, around ethics and preparation, again, this is working with ethical bodies in countries where vaccines may soon be tested. So the abbreviation spells out FRAP, and really what we're talking about is a pandemic preparedness strike force, using all those elements that I listed there. We're happy to partner with CEPI in this. Thank you.
Thank you very much. Thank you very much. So I'd now like to invite to the podium Professor Emanuele Montomoli, who is President and Chief Scientific Officer, of the Italian research and service company Vismederi. So Emanuele is going to give us his perspective from Italy and from within the vaccine development ecosystem.
Yes, good afternoon. Sorry. It's fine. Good afternoon, everybody, ladies and gentlemen. Let me thank Ministry of Health and Welfare of Korea for inviting me, and let me thank also IVI, WHO, and CEPI for this nice meeting. So, a couple of words about myself and about the company. So, my name is Emanuele Montomoli. I'm full professor on public health at the University of Siena in Italy. In 2009, we set up a company which is called Vismederi as a startup of the university, because at those times, several companies requested us to do analysis, serological analysis for preclinical and clinical trials at the university sites. So my rector told me that this is a pure commercial activity. It's much better to set up a company. The company grew up a lot. In the last 10 years, We enroll more than 300 employees now, and we have an annual revenue around $50 million per year. So we achieve a lot of quality certificates, of course, and we separate some sister activity, not in the vaccines or in the biologicals, but for drugs, for instance. So just to say that in 10 years we have done a lot. We have 2 buildings in Siena. One is the headquarters, which is in the downtown, and the second one is between Siena and Florence, is the laboratory park, where we have 5,000 meters square of labs, with the high containment labs as well, and this is the big advantage of us. We have 3 biosafety level 3+ lab that has been very useful for the CEPI collaboration and during the pandemic time. So what we are doing and which is our role with CEPI and with the public health engagement. We are not a CRO, we are a laboratory company, and we usually perform optimization and validation of analytical assays that has been used for clinical trials and for preclinical activities as well. We don't want to enroll volunteers, for instance. We don't have pharmacovigilance. This part is not in our focus. We are 100% focused on laboratory activities and, of course, in logistic and as a storage of samples, etc. So we have business almost everywhere in the world, and we get our results from both private sponsors. I mean, it's not confidential. We have master agreement with several big pharma companies as Pfizer, Seqirus, and I can say some companies in GSK and some companies in Republic of Korea as well. But we get project from no-profit organization as well, as for CEPI, PATH, the Gates Foundation, and several other. So the big achievement in the last 3 years is the release of the Pfizer-BioNTech vaccines. The December 27, 2020, there was a press release where BioNTech acknowledged us as well for the analysis done in the US and in Europe, and the day after was the first day of the first COVID vaccines in the world. This is what we are doing now. After that, in the April 2021, we were engaged in the CEPI network for the CEPI laboratory networks, together with 6 other labs in Europe and in other parts of the world. That's the first network of CEPI labs. Originally, we had 8 laboratories, not in Africa, not in China, not in Latin America. And we established 6 assays for the vaccines evaluation. At those time, we had 60 manufacturers who's waiting for analysis, and more than 20,000 sera samples in the fridge already ready to be analyzed. The situation, of course, increased, and in 2023, the labs becomes much more, and we have labs in the African continent as well. We have a lab in China, and very soon, I really hope to have a lab in Latin America as well. So, I already said that we established 6 assays for the COVID vaccines evaluation. Of course, the assays are focused to investigate the humoral immune response and the cell-mediated immune response. When we started with the lab network, each different lab has each procedures. We try to harmonize the procedure, and in the end, we agreed to have 2 ELISA assays for the evaluation of not functional antibodies, IgG antibodies, and we decided for the 2 ELISAs, one versus the spike protein and one versus the receptor binding domain. And we did also neutralization with a classical wild-type virus or with a pseudotype neutralization. And in addition, we set up a method for the cell-mediated immunity as well, just to investigate the production of interferon and interleukin after the stimulation with COVID. Of course, the lab network was standardized with international standard. The WHO international standard was established and were produced by NIPSC in the US. And originally, our experiment was done with the original ancestral 1 strain, but very recently, we have expanded to analysis of samples with several other variants of concern. We had a lot of experience in this field. We published, even before, in April 2020, the first paper on neutralization for antibodies response to the COVID vaccines. That's the first paper that we published. In 2 months, we got more than 500 citations. We set up the method in the lab, we validated the method, and we shared the method with the lab network as well. In addition to that, we standardized the pseudotype neutralization, because the question is that using the live viruses, we need high containment labs. Not several labs in the world has high containment laboratories. So for this reason, we decided to standardize let me say, it's not correct, but we usually use a false virus neutralization using the pseudotype platform. And that's exactly the role of, one of the role of CEPI. That's quite technical questions. I don't want to bother too much on that. But just to explain that before the CEPI network, we had several platforms, for instance, for the pseudotype neutralization. Some labs use the lentiviral pseudotype, other labs use the VSV pseudotype, and sometimes the results are not comparable. I was in a meeting during the COVID, and a company fight each other with the other because they have a higher titer obtained with the different assays. That's not good, of course. And thanks to CEPI, we have harmonized all these assays, and the pseudotype neutralization is one of the most important. So where we are now, the labs are 10 now, as I said before. The methods are still 6. The company which requests support are still around 50, and the sera samples analyzed are 70,000. 70,000 samples doesn't mean that 70,000 analyses, because as I said before, we have 6 assays. 70,000 are usually analyzed in several different assays, and sometimes for several different variants of concern. I mean, in the end, I believe that we have analyzed around 500,000 sera samples from clinical trials from COVID. And what the future? Of course, we heard a lot about, and we know a lot about 100 days ambitious objective of CEPI. And of course, we know that in the CEPI list of priority pathogens, we have Lassa, Nipah, Ebola, Marburg, and several other microorganisms. We already work on that. I heard before from Jerome that there's a good vaccine on chikungunya. We have neutralization for chikungunya. We establish a good neutralization for chikungunya, and we are ready to share these assays in the CEPI network and with other qualified labs. So, last 2 slides. Quality is very important, of course. When the labs was selected, we check very well the quality level of some labs, and where the quality level are not enough, we transfer the quality level appropriately for this kind of network. At least we need GCLP guidelines, and at least we need ISO 15189 certification. The last slide is that thanks to CEPI, I'm very proud about that, we do also capacity building and technology transfer. So in Siena, we have quite famous master in vaccinology and drug development, and CEPI, together with the Gates Foundation and several companies, support, and IVI, of course, support the master in vaccinology, the activities for the master in vaccinology. And thanks to the master in vaccinology, we have transferred the technology for the lab assessment in several rural areas in low-medium incoming countries where it was impossible to set up the method by myself. Thank you very much for your attention.
Thank you very much, Professor. So interesting to hear from somebody who's right in the network, and that vitally important lab network that was set up in the midst of the pandemic. Hopefully will be expanded to take on new members and new pathogens in the future. Our next 2 speakers are both from the pharma and biotech sector, but each comes from a different part of the world. First, I'd like to welcome Mr. Shadiq Askazia, who is the Chief Executive Officer of the vaccine manufacturing company Bio-Farma, which is based in Indonesia. So, welcome, and please let us have your thoughts.
Thank you. Good afternoon, ladies and gentlemen. This is a great honor for me to be part of this precision event. And thank you to Minister of Health of Korea, CEPI, and IVI. First of all, I would like to tell you about biopharma is, what we do, and what we align ourselves with global pandemic preparedness. Biopharma is a state-owned company which is produce vaccine and antisera based in Indonesia. Biopharma was established a long time ago. It was 1890 or around 133 years old. Our workforce about 1,500 employees. We implement an integrated quality management system with some certification, and within that could achieve WHO pre-qualification for many our product. Our product have been used in more 152 countries. Among this, 53 countries are member of Organization of Islamic Cooperation or OIC, and some of our product is already accepted in there. And 49 countries are from Africa. Our total annual capacity is about 3.2 billion doses per year. Here is the list of products that we produce at the moment. As we can see here, a list of vaccine products. We produce vaccine that needed for national immunization program in Indonesia. Until now, in Indonesia, we have 14 NIP as well as UNICEF program. During COVID-19 pandemic, we successfully developed a COVID-19 vaccine that has been distributed in Indonesia. The latest vaccine product that was licensed in Indonesia is typhoid conjugate vaccine. We collaborate with IVI for the development of vaccine. Thank you for IVI support. And we are so grateful that we had very successful collaboration with IVI, especially for TVC vaccine. And we are tremendously honored that the First Lady of Korea visit Biopharma to celebrate this great achievement. Collaboration between Biopharma and IVI for the success of typhoid conjugate vaccine, and the vaccine was approved for commercialization from Indonesia and RA or BPOM. Besides vaccine, we also supply antigen bulk for other vaccine companies. Sorry. Besides vaccine, we also supply antigen bulk for vaccine companies like polio, measles, tetanus, and so on. We also produce antisera, diphtheria, and tetanus antivenom. In addition, we also produce diagnostic kit, COVID-19 detection kit, and HPV. Lastly, we also collaborated with several vaccine companies to have partnership for several products such as MSD, Takeda, and others. Our products have been pre-qualified by WHO in this milestone of WHO PQ for biopharma product. Started 1997, our oral polio vaccine and measles, and more vaccine has been approved as well as since then. The product have been registered in more 15 countries and more 17 marketing authorization. And we are so pleased that in 2020, we got emergency use listing or UAA from WHO for emergency use for novel oral polio vaccine, and is the first vaccine listed under WHO emergency use for its NOPV2. And thank you for supported by BMGF. And during the pandemic, we contribute to provide COVID-19 vaccine for Indonesia. There are two schemes that, at that time, for short term, we collaborate with Sinovac to get immediate access to the vaccine for protect Indonesian population, and it was very successful. The second strategy that we developed in-house COVID-19 vaccine collaborate with Baylor College Medicine from USA. This vaccine has been approved under UAA Indonesia, and the development of vaccine took about 15 months. Total was distributed for 120 million doses with using system track and trace for delivery time and quality control. Just for your information, Indonesia have 7,000 active islands. It's not easy for our team to distribute to logistic and supply of vaccine. As we know that SHAPE with the great contribution during COVID-19 and actively lead on how and we could better be prepared for the next pandemic. One of the reasons for pandemic preparedness in 100 days mission, we are case of pandemic, the vaccine is hoped to be ready faster to be developed with 100 days. SHAPE initiative is joined to initiative in collaboration with vaccine manufacturer, including biopharma and the stakeholder. The main output is to create template document to be followed by manufacturer for vaccine development in case of pandemic. This is biopharma has capability in producing vaccine using well-known technology platform, live attenuated, inactivated, toxic recombinant protein, and conjugate vaccine. Lesson learned from COVID-19 pandemic is it important to require new technology platform like mRNA platform and viral vector. Biopharma is now acquiring modern technology through several collaborations globally. WHO has appointed mRNA technology transfer hub in South Africa, Afrigen and BioVac, and 15 countries as mRNA technology recipient, where Indonesia or Biopharma is one of the recipient countries. This is mRNA platform development program at Biopharma. If we look at the figure on top left, those our global collaboration with each scope collaboration. We have several collaboration with Manchester University from UK for R&D activities, WHO, MPP, or facilitator of IP throughout the development program. Collaboration with Afrigen and BioVac, South Africa, part of the WHO Tech Transfer Program. Collaboration with SHAPE for funding for mRNA development. And we also have collaboration with Quantum for co-development. This is timeline our mRNA development with COVID-19 as model. For disease model for vaccine development beyond COVID, we are still open for discussion with our strategic partner with some area of interest such as SHAPE priority list, endemic disease in Asian or other. Lastly, this is about the business continue plan of mRNA in biopharma, where antigen design will be done of our partners, process development in progress of biopharma, and deliver will be by both by biopharma and our stakeholder. This is construction. In this opportunity, we would like to appreciate SHAPE for supporting biopharma to approve funding for biopharma mRNA program. Along with support from Indonesian government, we are constructing infrastructure from mRNA manufacturing facility as shown here. We hope there is opportunity for collaboration and more funding from others like Asian Development Bank maybe. So this is strategy and significant support from WHO, MPP, SHAPE, and also Indonesian government, and our experience capability, Indonesia could be a vaccine manufacturer hub for pandemic preparedness in Asian region, which is critical for self-reliance during pandemic. Thank you for attention. Ganha Samida. Thank you.
Thank you. Thank you very much indeed. So we now have a distinguished speaker from this nation's pharmaceutical sector in the form of Ms. Jin Park, who is Executive Vice President of SK Bioscience, which is a major partner of CEPI's here in Korea, and was, of course, also the developer behind the Sky CoV-1 COVID vaccine, which was approved by the WHO. So, Ms. Park, the floor is now yours.
Thank you. So I think I am the only one who will deliver my presentation in Korean. So please use your translation device. 네, 안녕하세요. 저는 SK 바이오사이언스에서 해외 사업 개발과 퍼블릭 헬스 비즈니스를 담당하고 있는 박진선이라고 합니다. 사실 이 세션에 패널로 참가하고 계신 여러 그 패널 분들의 그 긴 경험과 또 커리어와 컨트리뷰션을 고려할 때 사실 제가 여기 자리에 적정하지는 않는 것 같은데요. WBS가 한국에서 되고 있고 그 다음에 또 SK 바이오사이언스가 그 한국에 처음 코로나 백신, 코비드-19 백신을 개발한 업체로서 또 크레딧을 받아서 이 자리에 서게 되어 크게 영광으로 생각합니다. 그리고 저희가 그 코비드-19 백신에 대해서 사실 10년 정도 이루어지는 것을 1, 2년 안에 발매를 했고 사실 그것만으로도 엄청난 어치브먼트인데 이걸 다시 또 3분의 1인 100일 안에 이루겠다는 세피스 100 days 미션 아래서 사실은 세피를 주도로 한 글로벌 이니시에이티브스와 각 정부, 저희 정부를 포함한 정부 레벨 그리고 IBI 같은 리서치 인스티튜트들 뭐 여러 각계에서 이를 달성하기 위해서 엄청난 노력을 하고 있는 것에 대해서 산업에 있는 한 사람으로서 리스펙트와 감사의 말씀을 먼저 드립니다. 사실 넥스트 팬데믹이라고 하는 것은 사업을 하는 기업 입장에서는 굉장히 불확실한 미래, 사업의 환경을 극복해서 만들어내야 되는 비즈니스입니다. 따라서 사실은 그에 합당한 리스크 미티게이션 플랜과 또 이를 단순히 회사의 미션만으로 진행하기 어려운 여러 가지 챌린지들을 가지고 있습니다. 그래서 이를 극복하기 위해서 한국에 있는 작은 회사인 SK 바이오사이언스가 왜 어떻게 또 이렇게 넥스트 팬데믹에 같이 참여해서 일을 하고 있는지 저희의 경험을 공유하는 자리로 가지겠습니다. 시작하기 전에 SK 바이오사이언스는 사실 20년 정도 백신 역사를 가진 사실은 상대적으로 굉장히 역사가 짧은 회사고요. 2008년에 연구를 시작한 이후로 14년부터 자체적인 제품들을 발매를 해서 글로벌 마켓에 공급을 했습니다. 그리고 동시에 사노피 같은 회사에 저희의 기술을 라이센싱 아웃을 해서 지금 현재 또 백신을 같이 개발하고 있고 사실 팬데믹 전부터 저희가 IBI와 타이포이드 컨주게이트 백신을 시작으로 해서 한 3가지 제품에 대해서 퍼블릭 헬스 쪽에서 콜라보레이션을 게이츠의 지원을 받아서 진행을 했고요. 사실은 이런 콜라보레이션이 저희가 팬데믹 이후에 이렇게 굉장히 많이 증가한 퍼블릭 헬스 콜라보레이션에 대한 문을 열어줬다, 기회를 제공했다고 말할 수 있겠습니다. 그리고 팬데믹 기간 동안 이런 콜라보레이션뿐 아니라 저희가 2개의 추가적인 코로나 백신인 아스트라제네카와 노바박스의 코비드-19 백신을 저희 공장에서 생산해서 또 글로벌 커뮤니티에 공급을 한 그런 기회가 있었습니다. 사실 저희가 지난 코비드 백신의 개발도 말씀드린 것처럼 엄청난 어치브먼트라고 생각을 합니다. 그래서 그것을 여러 리포트들에서 어떻게 그게 가능했는지에 대한 분석이 있는데 저는 오늘 이제 세피가 2022년에 발매한 리포트를 가지고 나왔고요. 사실은 mRNA나 바이럴 벡터 같은 기술을 미리 준비했던 것 그리고 그걸 생산할 수 있는 매니팩처링 캐패시티들이 사실은 좀 여러 가지 이슈들은 있었지만 저희가 가지고 있었던 것 그리고 무엇보다도 사실 1, 2, 3상을 한꺼번에 진행하고 또 그걸 빠르게 허가해주는 그 인허가와 임상에서의 이노베이션 이런 것들이 큰 역할을 했고 사실 그것이 코박스나 혹은 지금 이루어지고 있는 여러 가지 글로벌 파트너십이라는 큰 하모나이즈된 틀 안에서 이루어졌던 것을 여러분 모두 다 잘 알고 있을 것이라고 생각이 됩니다. 네, 뭔가 있었는데요. SK 바이오사이언스 사실 팬데믹 중간에 저희의 자체 백신인 그 GBP510 스카이 코비1 제품을 발매했는데요. 실제로 이 제품은 뭐 SK 바이오사이언스의 코비드-19 백신이라고 불리긴 하지만 실제적으로 저희가 기여한 부분은 어쩌면 굉장히 스몰 포션이라고 할 수 있을 것 같습니다. 게이츠 재단과 세피의 지원을 받았고요. 또 GSK AS3라는 명효성 강제, 그다음에 IBI가 임상 수행 자체 그리고 더해서 세럴러지컬 에세이, 그다음에 한국 CDC가 저희 자체적으로 진행한 헤테롤러거스 부스터 백시네이션을 지원하셨고 또 그 외에도 저희의 오리지널 테크놀로지는 오랜 기간 게이츠 재단의 지원을 받았던 IP IPD, 워싱턴 대학의 IPD에서 저희 쪽에 기술 이전을 해주셨습니다. 사실 이 외에도 여기 잘 언급할 수는 없지만 엄청나게 많은 미국과 유럽의 그 아카데믹 인스티튜트에서 저희의 임상 시험이나 전임상 시험들을 많이 지원해 주셨기 때문에 사실은 저희의 그 코비드 백신은 사실은 정말 지니윈이 콜라보레이션 프로덕이라고 부르고 싶습니다. 그럼에도 불구하고 이제 1, 2년 걸렸던 백신을 더 100일이라는 더 단축된 기간 안에 어떻게 발매할 것인가에 대한 사실 여러 가지 논의들이 엄청나게 액티브하게 이루어지고 있고 실질적으로 금방 세피의 제인께서 말씀하셨지만 이 앰비셔스하고 챌린징한 이 세피스 100데이 미션을 위해서 그것을 리얼리티로 만들기 위한 여러 가지 노력이 추가적으로 분명히 필요할 거고요. 과거에 했던 여러 가지 잘못된 부분에 대한 반성도 있지만 또 과거에 잘했던 것을 훨씬 더 강화하기 위한 여러 가지 노력이 필요하다는 측면들이 강조되고 있는데요. 동일한 리포트에서 결론적으로 기술, 저희가 특히 다양한 파토젠에 대해서 미리 연습을 해두는 것, 그리고 또 밝혀져 있는 백신 캔디데이트이 나오면 빠르게 그것을 임상 단계를 거쳐서 결과를 가지고 나올 수 있게 해주고 또 그것을 임상적으로 빨리 수행할 수 있는 인프라스트럭처가 갖춰지게 하는 것과 더불어서 사실은 기존 코비드 19 팬데믹에서 가장 이슈가 됐던 게 바로 equitable access에 대한 여러 가지 이슈들이었을 것입니다. 그러니까 선진 시장에서 매니팩처링 퍼실리티가 다 독점되었고 이로 인해서 사실은 저희가 소위 말하는 unprivileged country에 적정한 시점에 equitable access가 되지 않았던 부분들에 대한 반성을 기반으로 글로벌하게 이 어떻게 매니팩처링 캐패시티를 equitable access를 위해서 할 것인가 이런 것들이 해결해야 될 숙제들로 제시되었는데요. 사실은 저희 이제 SK가 그 가운데서 어떤 일을 하고 있는지 얘기를 하기 위해서 다양한 여러 가지 이런 키 아젠다들이 있지만 저거를 좀 간단하게 정리되어 있는 제가 이제 하나 레퍼런스로 가져왔는데 사실 크게 두 가지로 봤습니다. 하나는 diversified innovative technology를 사실은 여러 가지 파토젠에 대해서 미리 가지고 있는 것 부분과 앞서 말씀드렸지만 어떻게 매니팩처링에 대한 캐패시티를 글로벌하게 이퀴터블하게 할 수 있느냐 이 두 가지 측면에서 SK가 어떤 일을 하고 있는지 말씀을 드리겠는데요. 사실은 저희는 공정의 강점을 가지고 있기 때문에 사실은 기술적인 측면에서 많은 협력 그 기관들과 협력을 하고 있습니다. 사실은 하지만 그것만으로는 equitable access나 타임리한, sufficient한 supply는 힘들기 때문에 여기에 기재되어 있는 많은 그 퍼블릭 헬스 쪽에 일을 하고 있는 기관들과 협력은 너무 필수적인 것 같고요. 현재 그 기술에 관련해서는 사실은 세피와 여기 나와 있는 미국 정부뿐 아니라 유럽, 그다음에 앞서 발표된 저희 한국 CDC까지 다양한 방면으로 지원을 아끼지 않고 있고 mRNA 당연하고요. 그 외에 브로드 스펙트럼의 백신, 그다음에 기타 여러 가지 플랫폼 테크놀로지는 지원이 되고 있으며 또 mRNA가 가진 단점을 극복하기 위한 단백질 백신에 대한 그 지원도 되고 있습니다. SK는 현재 넥스트 팬데믹에 대해서 세 가지 프로그램으로 일을 하고 있고요. mRNA가 지금 게이츠 재단의 지원에서 FPA 지원으로 해서 사실은 우리가 모두 이퀴러블하게 쓸 수 있는 백신의 플랫폼을 만드는 그 연구를 하고 있고 또 판사베코 바이러스 백신이 저희 GBP510의 기술을 그대로 차용해서 판사베코 바이러스 백신을 개발하고 있으며 또 좀 독특하고 재미있는 컨셉의 트랜스미션 블락커인 제품을 게이츠와 이아비와 함께 연구 개발하고 있습니다. 그 다음은 바로 매니팩처링 캐패시티일 텐데요. 사실 여기 다 제가 설명을 드리진 않겠지만 아프리카, 유럽, 그다음에 mRNA를 어떻게 디스트리뷰션 할 것인가 차마 저희 이제 선진국인 유럽조차도 각 지역별로 필요한 플랫폼에 대한 매니팩처링 캐패시티를 가지기 위한 다양한 이니시티브가 돌아가고 있습니다. SK의 경우는 앞서 말씀드린 R&D와 매니팩처링 캐패시티를 하기 위해서 저희 나름대로 또 노력을 하고 있는데요. 2025년에 이동할 R&PD 센터에는 저희 자체적인 R&D 센터뿐 아니라 같이 일을 할 수 있는 오픈 이노베이션 랩을 만들어서 저희의 파트너들이 일할 수 있도록 하고 또 파일럿 GMP에서 저희가 새롭게 도입한 플랫폼 테크놀로지들이 바로 연구개발 목적으로 GMP로 생산할 수 있게 하는 기반을 만들려고 하고요. 그 외에 저희가 사실은 매니팩처링 관련해서 스카이쉴드라는 프로젝트를 소개해드리고 싶은데 이것은 사실은 각 지역별로 이런 백신 캐패시티를 가지고자 하는 여러 국가의 정부들과 협력을 해서 단순히 기술만 이전해주는 것이 아니라 저희가 팬데믹 동안 배웠던 여러 가지 생산에 대한 노하우와 그 다음에 이후에도 그 패실리티를 짓고 난 이후에도 지속적으로 롱텀으로 그 백신 엔티티와 인게이지해서 바이어블하고 서스테이너블한 비즈니스 모델을 만드는 사업을 하고 있습니다. 사실 저희의 에스퍼레이션인데요. 결론적으로는 사실 이 모든 흩어져 있는 여러 가지 비즈니스 엔티티가 모든 걸 갖출 수는 없다고 생각합니다. 저희는 이거에 R&D와 매뉴팩처링 네트워크를 만드는 게 저희의 꿈이고요. 사실 한국 회사가 한국에만 모든 걸 갖출 수는 없다고 개인적으로 저희가 믿고 있습니다. 그래서 이를 통해서 좀 더 효율적으로 저희가 허브에서 만드는 플랫폼들을 네트워크 안에서 쉐어하고 또 이를 통해서 각각 엔티티가 바이어블한 비즈니스를 만드는 것, 그것이 저희의 목표고요. 이 마지막 페이지인데요. 사실 결국은 넥스트 팬데믹을 위해서는 세트 플레이, 저희가 배구나 축구를 할 때 세트 플레이 연습을 하잖아요. 사실 그런 노력들이 정말 많이 필요하다고 생각이 됩니다. 그리고 이제 한국에 있는 기업으로서 마지막 말씀드리고 싶은 것은 사실 어려운 환경이었던 옛날에도 녹십자, LG, 유바이오로직스, 또 여기 SK바이오텍 많은 분들이 사실 인더스트리에서 많은 노력도 하셨고 또 저희 정부 얼마 전에 WLA로 승급도 했거든요. 정부의 많은 지원 그리고 또 같이 일하는 저희 아주 좋은 리서치 기반들 그리고 IBI 같은 기관 그다음에 라이트 재단 같이 게이츠와 정부가 재무적으로도 저희를 지원해주는 펀드들 한국은 사실은 여러 가지 면에서 봤을 때 굉장히 블레스드된 컨트리라고 생각이 되고 저희뿐 아니라 여러 인더스트리에 있는 많은 기업들이 다이그노스틱, 테라피틱스, 백신 모두에서 이 넥스트 팬데믹 프리페어니스에 같이 동참할 수 있었으면 좋겠습니다. 감사합니다.
Thank you very much indeed. Very important message there about practicing so that we are better next time. And finally, but by no means any less crucially, we welcome a friend and partner and colleague from Australia to talk to us from the point of view of academia. Professor Trent Munro is a researcher and developer, a vaccine developer at the University of Queensland and a specialist in protein biochemistry. His team at the University of Queensland have been working with CEPI for a number of years on a new type of vaccine technology that uses a molecular clamp to stabilize proteins. He's also, as it happens, mentioned in my book. So I hope you're going to read it on your way home. The floor is yours.
Thank you very much. And thank you also to Korean Ministry of Health and Welfare and CEPI for the support to be here today. So what I'm going to try and talk about today and get you excited about and sort of understand what we achieved during COVID was really how we can strengthen academia working with industry in our future battle against future pandemics. So if we think about drug development, as we all know, it's a very challenging, complex and torturous path. And if we think back into the biotechnology sector, there's this wonderful new podcast that was highlighted in Nature that gives some of the real war stories about what we were trying to do very early on. So when we think about the early days of monoclonal antibodies, there was decades of work that went in before success. If we think about the early days of RNA, in particular short RNA, companies spent many hundreds of millions of dollars trying to figure out how we deliver solutions for patients. So when we think about that, we really need to cast our mind back and think about how we make these early technologies more successful in a more rapid timeline. Obviously, during COVID, we were able to bend the curve and shift things and do things that many of us didn't think were possible. We think about the idea of developing this vaccine so quickly. It was really only the emergence of the collaboration that shifted the dial that let us bring these solutions forward. And of course, now we've got an even more ambitious goal with thinking about how we can do this in 100 days. So how do we better bridge the academic style, the academic benefits, the academic advantages with what industry brings in their ability to bring scale to the challenging problems we have to deal with? I think COVID was a good lesson. I wanted to highlight a few examples there and then tell you a little bit about our journey. So if we think about how quickly we moved during COVID, I think it's really great to cast our minds back. And it was interesting to me to go back and put this talk together. I mean, just how quickly we were able to see the first publications that highlighted the sequence of COVID-19 or the virus that causes COVID-19, how quickly we solved the structure of the spike protein, how quickly we identified the receptor and how quickly candidate vaccines were designed. So all of this happened essentially from February and March in 2020 after just learning about the virus for COVID-19 in January. So an incredible set of researchers were able to bring these solutions and data forward very quickly. If we then think about where we are today, absolutely amazing amount of data that we've generated. So more than 300,000 publications have been now published for COVID. And I was talking with a colleague of mine about this, and he said, well, let me just have a look what the total publication metric is for HIV over the last 40 years. And I think it's about 419,000 papers over 40 years. And here we are with COVID, just after a couple of years or a few years at 300,000, more than 300,000 publications. Absolutely staggering. When we think about the technologies that we use to develop these new vaccines for COVID, I think many of us also forget how quickly these discoveries flowed from being in academic settings through to commercial or late-stage products. Whether it was understanding how we do antigen stabilization for better immune response, whether it was the technology that led to the ability to decrease the innate immune response for mRNA, as was highlighted, which has now been awarded a Nobel Prize, or whether it was the types of technology we needed to harness for viral vector-based platforms of vaccines. All of these things led to incredibly rapid translation into vaccine products. So if we were able to now time shift ourselves back a little bit to 2019, which I know is a little bit difficult, seems like a long time ago, you know, CEPI had this slide where they really picked out their key pathogens of interest, and I think what they did was absolutely critical, was also have a pathway for disease X and think about the platform technologies we would need in this pre-COVID time. As they thought about that, they were able to then quickly deploy those Disease X programs into partnerships that were highlighted in the very early days of the COVID-19 outbreak, and the group that I was part of at University of Queensland was one of those groups. So I just wanted to highlight the people behind this technology. So Professor Paul Young, Professor Keith Chappell, and Professor Dan Watterson had really come up with a novel technology that CEPI thought had potential in the future of disease, of pandemic response. And really this was a technology that sits behind a recombinant protein-based subunit vaccine. Of course, what we understand is that to mount an efficient immune response, we need the right antigens. The team here recognized that the pre-fusion form of some of these complex viral antigens had an opportunity to elicit a more powerful immune response. The technology they developed here was really figuring out how to lock these particular antigens into that pre-fusion form, and then use that lock or this clamp also as the purification domain that would accelerate the ability to do scalable manufacturing. This technology was deployed across a large number of exemplar viruses, and it was really the broad applicability of this that led to the CEPI partnership. And the goal for us back then was really to think about going from pathogen identification to clinical material in a short number of weeks. It felt bold, but it's nowhere near as bold as I think what is proposed in the current 100-day mission. That for us was really thinking about bringing this technology for the first time into the clinic, and doing that with influenza, then moving on to MERS and then RSV, and then being challenged with CEPI for a Disease X program. All of that essentially went out the window with the emergence of SARS-CoV-2, and we turned all of our efforts to thinking about, can we develop an effective vaccine against COVID? Just to highlight how I think wild things were back in those days, this was our development plan. So this looks very different from a sophisticated set of slides or timelines that you see presented. But this is one of my favorite pictures from this time, because it simply outlaid everything that we thought we were going to have to do over this period. And it was really trying to put all of these very complex jigsaw puzzles that we've all been talking about over the last couple of days together and do them as quickly as possible. For us, it was doing it as an academic group and then figuring the path forward. I knew there were two things that were absolutely critical for us. One, who was going to be our commercial partner, and two, how we were going to get enough money through the door to do all of this work in parallel, and that was really the focus. So we turned that mud map into something that looks a little bit more organized, and what you'll notice for every single one of these were just discussions we were having at the time, there was backups. For every single decision we were making, there were other organizations we were thinking about talking to. Could we even access their technology? We really didn't know, but it was really this thinking about parallel paths that was critical as we were moving forward. As we matured, what we were able to turn this into was essentially a complex partner network. And this is really what I wanted to highlight, that it was this unbelievable collaboration between us as academics and our academic collaborators with a broad range of industrial partners that let us accelerate. And some of the key partners are listed on here. Some of them are now parts of the CEPI lab network. Some of them continue to be major manufacturers for the commercial vaccines that were established. So the other thing that we did, and many other groups around the world did this as well, is that we knew we needed to take more risk. Now, that's always a difficult decision to make. You're spending money up front, you're doing work up front, and you don't even know exactly how that's going to work out. The key work we did up front was really the process development and the scalable manufacturing, and we ran that alongside our earlier stage clinical trials to make sure that if those trials were successful, we could move forward. So as many of you will know, and some of you will be familiar with this program, it was an incredible set of achievements which really went from being able to do those first set of immunogenicity experiments, design a vaccine. We were able to move to our first human dose in around 5 months, and then to phase 3 clinical trial readiness in about 5 months. This really was one of the leading recombinant protein-based vaccine candidates at the time. We'd scaled manufacturing with our partner CSL to tens of millions of doses. But one of the things emerged was that the very advantage of our platform, this clamp that allows us both to purify the vaccine and also maximize our immune response, that actual sequence caused some diagnostic interference with rapid antigen testing for HIV. So there was a very difficult decision made in December to halt the program. But that's not the end of the story. The team has now gone back, redesigned the platform with a second generation clamp. The team's also just finished their second clinical trial with that re-engineered protein, and the results out of that will be out very soon, and we're very pleased with what that looks like. So lessons learned, academic and industry collaborations can be highly successful when there's a truly shared focus. Each team needs to understand when to lean in and when to hold back. Rapid technology translation is not without risk. We can shift the innovation cycle, and to achieve the 100-day mission, we'll need new breakthrough approaches. All right, and with that, I'll close. I think my time's up, and thank you very much. Appreciate the time.
Thank you. Thank you very much. We have a few short minutes just to have a little bit of a discussion, and I was actually just going to pose one question to 3 or 4 of our panelists before I ask Jane and Jerome, hopefully, to sum up. But I'm just going to ask a couple of our panelists about this 100 Days mission, because, of course, the theme of this panel is accelerating and strengthening global preparedness through the 100 Days mission. So I wondered whether I could ask Ms. Jin Park, do you think that the 100 Days mission is truly feasible? And can you think of an example of 1 or 2 things that your organization or company is doing that you think others should do to be able to facilitate and accelerate that 100 Days mission, to make it feasible?
Oh, okay. Wow. The first question is the most difficult question. I'm not sure I can answer the question, but yes, as I also mentioned in my presentation, this mission would be very, very ambitious and very challenging. So in particular, maybe even when all the stakeholders, including industry, works on the technology and manufacturing capacity, that may be possible. But actually, regarding regulatory pathway, would be quite big challenges because, without the scientific rationale, nobody can guarantee that any product can be approved. It's because it would be benefits for the big population, but at the same time, it could be very risky and dangerous if there is any specific scientific rationale. So that's why, you know, if I can choose the most typical piece of elements for this mission, maybe that would be in relation to the clinical development and the regulatory pathway as a kind of final step of the supply of the vaccines and therapeutics.
Okay, thank you, thank you. Professor Montomoli,
Yes.
would you take that question on and see, do you think that the 100 days mission is feasible, and or do you think there are, you know, what do you see as the main hurdles that need to be overcome?
Okay, I try to answer, but it's not easy. It's feasible, 100 days, of course, but for my point of view, it depends a lot which kind of microorganism we have for the next pandemic. I mean, in case that we have next pandemic for an H5 influenza, 100 days are more than enough because we know almost everything. We know the vaccine productions, etc. In case that we have a completely new virus, or even worse, in case that we have a completely new bacteria that can give an outbreak or a pandemic, that could be a critical time. 100 days are probably not enough. What we can do, because we have to suggest a solution as well.
Yeah.
Invest a lot in the new platform, in the new technologies. Before in COVID, we never know about mRNA. But there's not only mRNA. We have recombinant vaccines, for instance. We have new platforms that can increase a lot the production in a short time.
Thank you. Professor Monroe, what's your perspective? Are you a believer in the 100 days mission?
I am a believer, and I'm also, we had a chance to discuss this briefly before. I think one of the things we all did very, very badly during the emergence of the COVID, during the start of the COVID pandemic, was our predictions were all wrong. So our predictions about, you know, what would a vaccine work, our predictions about platforms of vaccines, our predictions about how fast we could go, they were all wrong. And so, you know, I think we have to be bold in our future predictions, and that's one of the things that I think is fantastic about the 100-day mission, because it's really challenging us to think about all of those aspects: supply chain, manufacturing, regulatory, technology platforms. So yes, I think it can be done, and I think we'll find a way to do it.
Thank you. And Mr. Shadiq, would you give us your perspective from Indonesia?
Yes, thank you. I think for CEPI goals for 100 days to find out the vaccine, is it possible if using mRNA technology? But maybe it is not only not find out the vaccine, but we have to know about the regulatory basis. It is for clinical trial, we need maybe, this is clinical trial, phase 1, phase 2, and phase 3. This is need time. And also from the distribution side, yeah, like I mentioned, like Indonesia have 7,000 island active. 1,000 is not easy to supply them. But in my perspective, we are very support that CEPI have a goal like that. Yeah, this is how to accelerate to find out the vaccine, and we have maybe for standardized acceptance from regulatory when pandemic situation. So we can go faster. And the last, this is openness and willingness for the stakeholder to share information and given transfer technology for also global consensus and to control pandemic. Better for humanity, global health, not only for the business.
Yeah.
Thank you.
Of course. So collaboration and working together through partnerships is going to be one of the vital things to increase our preparedness for this inevitable emergence of a new disease X, but hopefully would make sure that another pandemic is not inevitable. I'd like to ask Professor Halton, Jane Halton, to just give us a few words of summary from this panel. Have you been... Has your message of hope been reinforced?
I hope so, and I think so. And can I make a couple of comments? Firstly, I hope everyone has heard what I think is a really interesting and clear story from across the panel. We've heard from people who work on research. We've heard from people who actually are in labs, who produce vaccines. I could go on. And all of them have talked in a way which I want to describe as this: practical, pragmatic, methodical, and essentially ambitious. And it's all those things which will get us to the 100-day mission. It's understanding we need all of these things lined up. We need everyone to work together, and as I said in my remarks, we need partnership, we need collaboration, we need goodwill, and we need good science. And I think that's what we've heard here today. And I hope many of you have heard from people you had never heard from before. Because the other thing we've learned is there are people doing this work around the world, and that is the network of people we need to engage.
Thank you. Yes, I have found it, and I hope the audience has found it too, fascinating to see how many overlaps there are. There are so many partnerships, and it's just, they don't just involve two organizations or two people, they involve multiple organizations, and quite often they overlap. And I think that's been a lovely thing to see here. Dr. Kim, could you give us your thoughts on the hope for the future?
Yes, so I think the panel was fascinating. And it really, to summarize it, really there are three elements: preparation, response, and partnership. And many of the comments actually fell into these big buckets. But under preparation, and we didn't emphasize it enough, but funding is very important. Funding to do the 100-day mission, but the importance of the research that was underneath all of the work that we could do very quickly was really critical, and that's something really only governments put a lot of money into. Philanthropic organizations maybe, but and some NGOs and IOs like CEPI. But really, this is a government function, and the importance of that research can't be underestimated. The other platforms for vaccines and process, and then the importance in preparation of practice. And I think Jin Park emphasized this, being able to do this through and through once. Then around responses, this is about testing in animals and humans, this is about approvals, and then finally it's about policy and uptake. And we often leave that part out, but it's the part that if we mess up, none of this is going to be effective. So a very important part. And the final part, and I think many people mentioned this, is the importance of partnership. Around for instance, the Korean government's response, which involves both public and private and international organizations. I think that's a kind of example of how we put everything together. But around the manufacturing partners, around international organizations, non-governmental organizations, and philanthropies. If you look at the at SkyCoV-1 or the work that Visma Dairy did or the work that University of Queensland did, it was really very important partnership between all these organizations that gave funding and put people together in the right situation to make them move quickly. And so, I think there's reason to be optimistic. I think we have a lot of the pieces identified, and really it's trying to put it all together and making it work quickly.
Thank you. Thank you very much. So I need to bring this session to a close now, so I'm going to hand back to my colleague MC. Thank you very much.
Thank you very much for that in-depth discussion. Ladies and gentlemen, once again, let's give a big round of applause to our honorable panelist and moderator. Thank you. And we do have one last photo shoot. I think we need to remove our chairs and desk. Can you please stand up and be on the stage? I think we can just take a photo together. So please be at the center of the stage. Can you please step a little bit towards your left-hand side? Thank you. If you don't mind, can you please take 2 steps forward? Take 2 steps forward. Thank you so much. Let's take a photo shoot together. Thank you very much for your presence once again to all of our participants. Ladies and gentlemen, let's give another big round of applause to all of our panels. And thank you, our moderator, for leading the session. Thank you. Now please return to your seats. Thank you very much. And ladies and gentlemen, we will be conducting the closing ceremony shortly. I'd like to ask all attendees, please take a short break. We will soon begin the closing ceremony in about 10 minutes, starting from 5:30. So once again, thank you, and see all of you at 5:30 here at the same venue. Thank you.